the abridged version of advanced glycation end products and the pathophysiology of angiopathies in type 2 diabetes.
Diabetes is a disease of altered chemistry and metabolism of both carbohydrates and lipids. Normally, the absorption of carbohydrates stimulates insulin secretion in the translocation of glucose transporter 4 from muscle and adipose tissues intracellular compartments to the cell's surface, in order to increase cellular uptake of glucose.
The hormone insulin acts as a second messenger that activates several cell signaling pathways, from the cell's membrane to the transcriptional component in the nucleus.
However, insulin resistance in type 2 diabetes is a post-binding abnormality in which skeletal muscle and adipose tissue's normal response to insulin is interrupted.
Consequently, glucose transporter 4 remains in the cell's intracellular compartments, and the in return causes an increase in the amount of glucose in the blood.
Due to muscle and adipose tissues' insensitivity to insulin, the pancreas secretes more insulin in efforts to maintain blood glucose levels, while adipose tissue releases free fatty acids. The net result is elevated fasting blood glucose levels and increased triacylglycerol synthesis in the liver.
Hyperglycemia bathes every cell type throughout the body. Consequently, what we see in type 2 diabetes is that cells that are unable to decrease the rate of glucose transported inside the cell become damaged.
We see this specifically in 3 cell types: the capillary endothelial cells in the retina, the mesangial cells in the renal glomerulus, and the neurons and Schwann cells in peripheral nerves, collectively representing microvascular complications evidenced in diabetes, retinopathy, nephropathy, and neuropathy.
However, altered lipid metabolism may play a leading role in the development of macrovascular complications, the leading cause of morbidity and mortality in diabetes.
The increased flux and oxidation of both glucose and lipids has 2 consequences: the increased formation of intracellular reactive oxygen species, and the increased formation of advanced glycation end products.
An increase in intracellular reactive oxygen species compromises the cell's natural antioxidant defense system.
The formation of advanced glycation end products, however, have a more deleterious effect cell structure and function.
First, AGEs, or glycotoxins, are modification of proteins and lipids that become glycated and oxidized through a non-enzymatic reaction with reducing sugars. Reducing sugars are monosaccharides such as glucose, fructose, and galactose, whose functional carbonyl group is either an aldehyde or ketone.
The formation of AGE spontaneously occurs when the unpaired electrons from the amine group, which are found on proteins, lipids, and nucleic acids, are transferred to the carbon of the reducing sugar's aldehyde or ketone carbonyl group, creating a temporary Schiff base and the amidori product, which is then oxidized to form an irreversible AGE.
AGE formation occurs naturally in the body as a part of the aging process. However, its formation is accelerated in diabetes due to the increased availability of reducing sugars.
Once formed, AGEs may bind to its receptor on the cell membrane, or glycate extracellular proteins.
Consequently, AGEs permanently alter the cell's structure and function, resulting in blood flow abnormalities, vascular permeability, angiogenesis, capillary occlusion, vascular occlusion, pro-inflammatory gene expression, and increased production of reactive oxygen species.
As the formation of AGEs increase, they accumulate in both serum and in skin.
Elevated serum accumulation of AGEs has been evidenced in peripheral and autonomic neuropathy, atherosclerosis and myocardial infarction, and precedes early signs of renal disease. It is also considered nephrotoxic, alters renal hemodynamics, and stimulates inflammatory processes, not to mention that it has been demonstrated to be a strong predictor for heart failure.
Likewise, skin accumulation of AGEs has been demonstrated to correlate with degree of neuropathic foot ulceration, an independent predictor for progression of microvascular complications, an independent predictor of acute myocardial infarction, delays wound healing, and may predict risk of future diabetic complications independent of hemoglobin A1c.
Clinically, since the accumulation of AGEs are dependent on the half-life of the protein, blood and urine samples of AGEs may not reflect levels of AGEs in tissues. Up to this point, accelerated AGE formation and accumulation in diabetes has been demonstrated to activate inflammatory and pro-oxidant mechanisms, impair cell signaling pathways, accumulate inside and outside the cell, and alter cellular structure and function, apart from overnutrition or genetic susceptibility. In addition to AGEs that are formed endogenously, the modern diet represents an important exogenous source of AGEs. Most notably, diet-derived AGEs analogously contribute to the whole-body AGE pool and AGE-related disease pathologies.
Exploring the formation of AGEs in various types of foods, including suggestions on reducing these glycotoxins in the diet, are described in the section, Dietary Advanced Glycation and Products.
The end of the abridged version of Advanced Glycation and Products and the Pathophysiology of Angiopathies in Type 2 Diabetes. Detailed explanations of various mechanisms involved in the development of vascular complications are available in the full-length version. Additionally, there are 2 other related videos, Overview of Diabetes and Vascular Complications, and Dietary Advanced Glycation and Products. This information is also available at the provided URL.